at genetics a little bit more in there with Mendel and the gene idea. Mendel was the one who did the studies with the pea plants in there to first study genetics. Back in the 1800s, they thought they used this kind of blending hypothesis that they thought your genetic material was the mix of two parents and your traits mixed together like paint. So basically, you know, if you had a yellow parent and a blue parent, you were going to be green. And that's not the way things actually happen. With the particulate hypothesis, which ended up being what they used for the gene idea, was that the parents passed discrete inheritable units that retain their separate identities in the offspring, and those units ended up being genes. So when Mendel did the studies of this, he studied pea plants in there, and he chose to track characters that only occurred in two distinct alternative forms in pea plants. So the character is a heritable feature that is going to vary among individual flowers. So something like a flower color in there. And the trait is the variant for each character in there. So that would be the different colors that the flower could be. So say purple or white in there. And it was important that he used the things that only had two distinct traits in here. Otherwise it would have really, he probably would have eventually come to the same conclusions in there, but he would have been a lot longer getting there if he wouldn't have started with something that had just the two options for the traits in there. So when something is considered true breeding, it's a variety that's only going to produce the same variety as the parent plant after self-pollination in successive generations. So another thing is, it was important that he studied this in plants, because you can't self-pollinate yourself as a human in there, probably a good thing. And only certain plants can self-pollinate too. There's some plants that actually have to have another plant around to get the pollination from something else. So the pea plants could self-pollinate, and it didn't take a whole lot of time for them to grow either. So hybridization is the mating or crossing of two contrasting true breeding plants. So if you had the two varieties, you would cross or breed two of the true breedings that would only produce the one type of variety in there. So the P generation is your parental generation. That would be those true breeding plants that you started out with. And the F1 generation, the F comes from filial, which means son. Why they decided to make these sons when your chromosomes are daughters, who knows, but your P's are sons in there. And so the F1 generation was the hybrid of the offspring from crossing the true breeding parents. And then the F2 generation was allowing the F1 hybrids to self-pollinate in there. So there is a whole lot of inbreeding of these plants in there to be able to test this. And with plants, it's not such a big deal. They can't walk around to find new mates in there. It doesn't have the same implications that it does for people or animals doing that kind of thing. So just because you read about it in here doesn't mean go home with brother and sister and get busy. So this diagram here, would somebody mind grabbing me a stick from the back? Thank you. I'm going to have to find a nice branch to use. A what? A window open? No, they don't open. It won't be long. Next quarter, I'm going to grow it in the chemistry room. Nothing's growing in there now. It's just incubators really stink. And I just didn't realize the ventilation would just keep it stuck in here so much. So next quarter, I'm going to grow it over there. And they won't know the difference or where it came from. And then my room will smell pleasant. It won't matter if they don't open the door. So you've got the parent generation here. This plant with purple flowers, if you crossbreed it with itself, it's only going to make purple flowers. This one, if you self-pollinate it, only makes white flowers in here. So the F1 generation, when you cross these two, all of them had purple flowers. Then when you crossed 2 within this generation here, you got 3 purple, 1 white in there. So the law of segregation came out of this in there. And so if the blending model was correct, all of those flowers would have been light purple in there. Instead, you got white and purple. So that threw out the blending model in there. But what you end up with is dominant and recessive traits in there. And so the dominant trait occurs at a 3-to-1 ratio with the recessive trait in the F2 generation in there. So when you look at the law of segregation, you've got the alleles, which are the different versions of the gene. So you have a gene for purple and a gene for white in there. And the alternative versions are going to account for the variations in there. So for each character, the organism inherits 2 copies of a gene, 1 from each parent. And if the 2 alleles at that locus differ, then the dominant allele will determine the appearance of the organism in there. And if the 2 alleles, or you're going to get the 2 alleles for a heritable character, they're going to segregate during gamete formation and end up in separate gametes. And so we say it's the law of segregation. They have a 50-50 chance of ending up on each gamete in there. So if we look here at those flowers again, so the purple flowers, they are going to be capital P's in there. So when you're doing this with the genetics, the dominant trait gets the capital letter. And you'll use the same letter for both traits. It's just one's going to be a small letter, one's the capital letter in there. A lot of times you go with the letter that's of the color or the trait that is the dominant one. So that's why the white ones are small p's in there. So the gametes, this one has no choice but to produce gametes that have a big P. This one has no choice but to produce gametes that are a small p. So when you put those together, what it's going to do is make the purple flower. So when you make this generation, each of these, half of their gametes are going to be the big P, half are the small p. So when you create the Punnett square of them, you take the eggs, each egg would have either a big P or a small p, and each sperm would have either a big P or a small p. What you put in the square is what you take from this one and this one. So each square has 2 letters on there. That's where a lot of people go wrong on the Punnett squares, is they'll just want to put 1 letter in each box. It's got the letters from each of the gametes that are in there. So 2 big P's, you're going to get purple. 1 big P, 1 small p, you still get purple. 1 big P, 1 small p, you still get purple. 2 small p's, you're going to get white. So that makes sense how that's done on the Punnett square? Okay. So the Punnett square is just a diagrammatic device for predicting the allele composition. And before I forget, you're going to want to know how to do these Punnett squares. And you have plenty of opportunity to practice doing these Punnett squares on here. In Canvas, there's this genetic practice links in there. If you open that up, these are all links that have worksheets or websites that allow you to practice doing the Punnett squares and the things that you would have to do in this section. Yes. You're going to want to know how to do these Punnett squares. Those practice sheets are there for a reason. And not because I want to see them. So the Punnett square allows you to predict this. So when you look at this, if this flower is purple, unless we already know it's a true breeding, we have to assume it's either got 2 big P's or 1 big P and a small p. This one, white, we know that has to have the 2 small p's because we know that's the recessive trait. So when you cross these 2, you have 2 possibilities. So if we want to know what the genetic makeup of this one is, we have to breed it and take a look and see what is the ratio of what you get for your offspring. If you get all purple offspring, you know it was big P twice. If you get a 50-50, you know you had one that had the heterozygous on there. So homozygous means the organism has identical pairs of alleles for the character. Heterozygous means it's going to have two different alleles. Your phenotype is what the organism's appearance is, or your observable traits. The genotype is your genetic makeup. So a test cross is what you do when you have an unknown genetic makeup and you want to know what you have. So a test cross does have limited usefulness in there. Great to do a test cross if you have a pea plant. However, if you look and you've got a human being, you don't say, well, let's just mate with him and see what his genetics are. Bad idea in there. You just don't do test crosses with humans. So monohybrids are where the parents are heterozygous for one particular character, followed in the cross. And monohybrid crosses are going to be where you cross organisms heterozygous for just a single character. So if you're looking at a dihybrid cross, then you've got two characters you're looking at. Trihybrid cross, 3 characters. Thank you. You can go on beyond that, and you can do crossing for 4, 5, 6, 8 characters if you want. However, nobody does that. I don't think I do 3 because they get really ugly to grade. It becomes very tedious for me to deal with more than 2. I really don't even like doing 2, but you need to know how to do it. So the dihybrid, you've got 2 characters that are being followed on the cross. So we're going to look at an example of those. And it's going to follow the law of independent assortment that basically says each of the alleles assorts independently during gamete formation, as long as they're on different chromosomes. Why don't we just turn it off? What was it on? How was it set before?
I don't know. I just turned the power to the light.
I have no idea how to program it either. It was on when I got into this room, and I never changed that. I don't even know where the manual is to program the thing. I'm not good with manuals. So when we look at this, if we have, this is looking at smooth peas and wrinkly peas, yellow peas and green peas. So here it's taking one from your pea generation that is yellow and smooth. Here the other one is green and wrinkly. So when you look at that, the possible gametes that you're going to have from this one, Y and R. For this one, or big Y, big R, small y, small r. There might be. This is only 2 traits in there. So with you could do individual for each trait and make a whole bunch of boxes in there, but instead what you do is, being that we know that it's independent assortment, you don't want to mess with the little boxes on this. You're going to do the big box that has 16 in there. So 1/4 chance of this gamete, 1/4 chance of this gamete, 1/4 chance of this gamete, 1/4 chance of this gamete. So it's a 1/4 chance because there's 4 gametes and you're only going to get 1 of the 4 in there. So when you do the cross, you've got big Y, 2 big Ys, big R, 2 big Rs. When you cross these 2 big Ys, big R, small r in there. 1 big Y, 1 small y, big R, big R. So does everybody understand how you get what goes in the box from there? The what? Yeah. You basically, you bring these letters into the box and these letters into the box. You just have to remember, bring all the letters into the box in there. So when you do that, your phenotypic ratio would be 9:3 to 3:1 in there. So if you didn't want to do these and you wanted to be able to do multiple crosses in there, some hybrid cross or something crazy like that, you could use math to do this with the laws of probability.
So the ratio is like 9 is like the number of what, yellow? And then 3 is the number
Yellow and
green
smooth.
and smooth, and then 3
Yeah, so yellow and smooth, yellow and wrinkly, green and smooth, green and wrinkly, like that. So if you wanted to do it using math, you would use probability. And I got to explain to you that you can do it with the math. I can tell you you're not going to have to do the math in here. We already know I don't like math. So probability ranges from 0 to 1. 0 means it's absolutely not going to happen. 1 means it absolutely is going to happen in there. So when you do this, if you're using the multiplication rule in there, that's used when you're looking to determine the probability of two independent events occurring together. So you multiply the probability of them. So if you have a 1/2 chance and a 1/2 chance, you multiply that, you get a 1/4 chance. The addition rule is the probability of any one of two or more mutually exclusive events that will be calculated in there. So you add their individual probabilities, like 1/2 and 1/2 gives you 1, or 1/4 and 1/4 gives you 1/2 in there. So this is just showing some examples in here of how you could calculate probabilities of this. And I'm not really concerned about you being able to do the math of it, because outside of doing it for a textbook like this, I've never had to use it for anything. If you understand the idea of how the genes pass through, that's more important. You could always, worst case scenario, make yourself a giant grid box and work it out that way. But I just don't see you having to do that either. So some things are going to have incomplete dominance in there. So complete dominance means you're going to get all of one trait or the other. The incomplete dominance, somebody probably saw red, white, and pink flowers at some point and came up with that blending hypothesis in here. But the particulate hypothesis can actually explain that as well. So here, when you have something that's got incomplete dominance in there, the way we indicate the traits, you're going to use a little superscript. So you've got CR for color red. That's how you know when you're looking at the gametes being crossed, is this going to have dominant and recessive relationship, which is what it normally has in there, that you just have the single letter. If it's got incomplete dominance in there, you use the superscript on there. So you have the color red, color white. So the gametes you make, CR and CW. So in your first generation, you get a CR and a CW together, and red and white mixed together to make the pink. So the gametes, half of them will be CR, half of them will be CW in there for the F2 generation. So you're going to get one that's red, two that are pink, and one that's white. So for codominance, here you can have two different alleles that both affect the phenotype in a separate distinguishable way. The example that we have for this is human blood types in there, the ABO blood types. So what that means is if you are blood type A, it means your gene type, you either got the type A or the type O, which has no marker, protein marker on it in there from the parents. So you either got both parents gave you the A marker, or one gave you the A marker, one gave you the O marker And what you will put on your blood is the marker for A, that A protein. Where you run into problems is you will make antibodies for the B protein that you don't have. If you're type B, you got B from both parents or B from one, O from the other. You put the B marker on your cells, and you'll make antibodies that would attack the A protein that you don't have. If you are AB, you got A from one parent, B from the other, you've got both of them on the surface, so you aren't going to make either one of the antibodies or you'd attack your own cells. And if you're type O, you got O from both parents, you don't make any markers, but you make antibodies towards both types in there. And so that's why the type O blood type is so useful, because this doesn't have any markers, so you can put it in any of these people, and their antibodies don't have anything to attack. However, you stick any of these in this person, and their body will attack it. Oh, it's okay. You can keep using it for now. So the dominant allele in there that's seen in the phenotype doesn't interact or suppress the recessive allele in there. So one example of a disease that's a hereditary disease is Tay-Sachs disease. With this disease, the brain cells can't metabolize certain lipids because the enzyme doesn't work properly. Then you build up the lipids, you get brain problems, seizures, blindness. Eventually it kills you. So at the organismal level, you have children that have 2 copies of the allele have the disease, and it's qualified as recessive. They've got 2 recessive traits in there. The activity level, if you have a person that is heterozygous, that got 1 good gene, 1 of the bad genes in there, because of the incomplete dominance, they will actually have a little bit of the enzyme in there. And so they can actually produce enough to be functional, even though they do have some dysfunctional enzyme in there, and they're still a carrier in there. So most genes are going to exist in more than 2 allelic forms in there. And so pleiotropy is where you have multiple phenotypic effects in there. So a lot of times in humans you'll have pleiotropic alleles responsible for multiple symptoms with certain hereditary diseases like cystic fibrosis and sickle cell disease. So pleiotropy is a gene affecting multiple phenotypes in there. Epistasis is phenotypic expression of a gene at one locus altering a second locus in there. So the example that is used with this is with lab dogs in there. So I think that this is a perfect excuse why I need to have a nice lab dog that's the classroom dog in there, that we could talk about breeding it in there. Yeah, those anti-dog people here aren't going to go for it, though. So I don't know what his disability is that he gets to claim to have that. Probably the only disability I could have would be some sort of like personality disorder, like I'm a witch.
I have anxiety and I need my dog. We're just put a vest on him. That's what my aunt does.
My dog gives me anxiety. So if we look on here, with the labs in here, you've got one gene that's going to determine whether or not they are going to deposit pigment in the hair. And then you've got another gene that's going to determine the color of the pigment in there. So the gene for pigment deposit is epistatic to the gene that codes for black or brown in there. So the genes are going to code for black or brown in there. So if you have the code for being a black dog, but you also get the gene for not depositing pigment, you're going to end up yellow in there. So what's going to override it is the first gene, or this gene in there that determines are you going to deposit pigment or not. It doesn't matter what color pigment genes you have. if you have the gene that says you're not going to deposit the pigment in your hair. So if you have the brown gene in there and it says you're not going to deposit pigment in your hair, you're still going to be yellow. So what makes a yellow lab is that they don't deposit the pigment in there. So that is how you can have various labs breeding, 2 different labs, and you get a mix of the puppies in there. And so the chocolate lab or the brown lab is the one that you'll get the fewest of in there because of the recessive traits in there. But it seems like you see black labs everywhere, and you're going to get more black labs than the yellow and brown labs together. So with polygenic inheritance, here you have the additive effects of 2 or more genes on a single phenotypic character. So it's the opposite of pleiotropy in there. And an example of this one, you've got quantitative characters in here like skin color and height in there. So that's how you can end up with children that are going to be skin colors in between 2 different parents in there, that will have heights in between 2 different parents in there. Is it can be kind of a blend in there, and you can have multiple genes affecting it in there. So then we have genes that are also going to depend on the environment. So the nature versus nurture in here, there's going to be exceptions where the environment can end up changing the genotype. And so when I was doing that continuing education last weekend, one of the things in there was looking at studies with the mother being obese during pregnancy, that it can actually change the genetics and make the child more likely to be obese because mom was obese during pregnancy. And you can actually have the genetics be influenced even the generation before with the grandmother being obese. And so is what? There's hereditary influences on there. So, I mean, it doesn't mean just because, like in my case, come from a long line of fat people. They're all fat. And so, you know, the nature in there is the genetics tell me I want to be fat. My head up here says I don't want to be like them. I don't want to be like them. However, there's still the genetics that are working in there, but you also have the environment that I grew up in where if there was candy, they were all fat, so you fought for it, because if you didn't fight for it, you weren't going to get it. So, you know, nature versus nurture, I got lots of things pointing towards being fat in there to overcome. So the norm of a reaction, a genotype is generally not associated with a strictly defined phenotype, but rather a range in there, and due to environmental influences. So they're looking at it, I mean, they're studying it with obesity, because there you can kind of look and you go, okay, this is pretty easy to see. Mom is above average weight on a scale. You can measure that. And a certain number of years later, I mean, they're looking even at childhood obesity in there. And we know with childhood obesity, most kids that are obese end up being obese adults in there. It's pretty easy to follow that and study that over the years. But there's also the possibility of saying, okay, if you are in this kind of environment, you will change your genes to be more likely to get cancer. However, it could mean you're more likely to get cancer 50 years down the line. That's a lot harder to study in there. So getting that information and getting it in a good scientific manner is, you know, a lot of times people say, well, why don't they just study that? Well, you know, one person can start the study, but it's going to have to be carried on through multiple generations in there to actually get the result. You could start the study and then have somebody else in the next generation know to benefit your grandkids. It's not going to be a quick answer in there. Okay. Thanks. So with the Mendelian patterns of inheritance in humans, since you don't just do a test cross and say, I want to know what your genes are, let's make a baby and see in there. Instead, we look backwards and do a pedigree on there where you look at the collection of the family history for the trait, and then you assemble it into this tree. So there is practice ones of these to do as well on here, and it can help calculate the probability of having a future child that would have a particular genotype or phenotype in there. And these here, doing the pedigree is actually worthwhile understanding how to do this, because say there is a lot of a certain type of cancer that runs in your family. If you see a genetics counselor, they're basically going to do these on you to see, okay, are we justified in doing genetic testing in there? Without having the genetics counselor document it this way, no one else is going to pay for that for you. No insurance will pay for it without that kind of documentation. The pedigree in there, of looking at family history in there to have it covered. And even then, it may not be covered by insurance, but the insurances that do cover it, that's what they require in there. So like for me, they wanted to do some testing in there, but we needed to know some information from a couple of my aunts who chose not to go get any testing done in there. It would have been covered for them, but they're like, well, no, I'm not going to do that. You got to remember, I got relatives that still think smoking's good for you. So they were not going to do that. So for me to get the genetic testing done, it would have been paid for out of my pocket. So I, of course, opted not to, like most people in there. But that's what this is useful for. So that's why you would want to be able to do that and be able to figure that out, is it helps you to be able to determine, okay, do I have a risk for this in there? Now, if you're adopted, you don't have that to work with in there to help you, but sometimes it's kind of nice if you're looking for future generations and you're going, well, I'm looking for me and my spouse, what are there things that I should be watching for in my child as a result of this? So when you make one of these in there, you've got male square, female round, and a person that's affected in there is going to be colored in. One that's not affected is not colored in. We don't necessarily know what their genetic makeup is in there, remember. All you can do is look and say, do they have the condition or not? Or do they have the trait or not? Yes or no, like that. So when you have a mating, the lines are next to each other. When you make offspring, the lines come down from the top. So one of the things I find in here when people make their pedigrees, if you do this, you are sick. You belong in the deep south kind of thing in there. So none of those lines in there. Remember, there's a difference between offspring, siblings, mate. Siblings, mate. There is no equal sign in between. So just, be aware of that when you practice making them, looking where the lines are in there for it. Yeah. So with recessively inherited disorders, heterozygous people are typically going to have a normal phenotype in there, and because only one of the alleles produces enough change in the specific protein in there, the disorder only shows up in the homozygous individuals. So that's how you end up with carriers, and a lot of times they don't know they are a carrier in there unless they look at a pedigree of their family going back, and then they can sometimes say, well, this is the odds that I am a potential carrier for this in there. So generally genetic disorders are not evenly distributed among all groups of people. So you've got different genetic histories in times when we were more geographically and genetically isolated in there. So it's only been relatively recently that you've had people that would go in between here or Europe or Asia, different countries like that, with air travel in there. So consanguous matings, that's the inbreeding in there. So when you look at it with dogs in there, a lot of purebred dogs have more of this going on because somebody wants to make the puppies in there. And I don't know what goes on in people's minds that they go, well, it's okay to do this with the dog because the dog is willing to, if it's in heat, anything that is a male dog is willing to give it a try. They're not real picky. But you do end up with a lot of the traits getting passed down in there. You have the traits occurring far more frequently than they should. So a lot of dogs, you could say certain breeds have tendencies to have like hip dysplasia and things like that because people do this in there. And even if you get a pedigree on dogs, I've known people that breed dogs that will lie on the dog's pedigrees that they submit in there. So it's no guarantees in there. And a lot of times with the genetic disorders, you can't tell until the dog is an adult, full grown in there that, yeah. So, to say, oh, you had the puppy certified for hip dysplasia, it doesn't really mean anything. Puppies don't get hip dysplasia. So some examples of these diseases, cystic fibrosis is the most lethal or most common lethal genetic disease in the United States. 1 in 2,500 people of European descent, or 4%, are carriers in there. It's more rare in other groups. The normal allele codes for this protein in there, FRO, that transports the chloride ion between the cellular fluid in there. And so the channels are defective or absent in the recessive homozygous person in there. So what it means is they make mucus that is thicker and stickier than a normal person. So it will build up in organs that create secretions. So pancreas, the lungs is a big one. A lot of people with cystic fibrosis, a lot of lung problems. Gastrointestinal tract, other organs. The survival rate has improved in there with them now. Most will survive into their 20s and 30s. So I know somebody who's in their early 40s with it, and he just had a lung transplant. Most of them do not have children. Their sperm have to swim through jello instead of through the ocean in there, so it doesn't really work well for that. Sickle cell disease in there. This is more common in people of African descent, 1 in 400 African Americans in there, and it substitutes a single amino acid in hemoglobin in there. The homozygous person will have all their red blood cells be normal in there. Low oxygen content in there can cause red blood cells to become sickle-shaped in there and clump. And so the heterozygous carriers have what they call sickle cell trait, where they are usually healthy and normal, but they can manifest symptoms when there is prolonged reduced oxygen in there. So 1 in 10 African Americans have sickle cell trait in there. One of the reasons why you see so much of it in that population is in countries where malaria was really prevalent, the heterozygotes have reduced frequency and intensity of malaria attacks. So it was made you more likely to survive malaria in those areas. So dominantly inherited disorders. You know what? We'll just stop on that for now.